Hello, everyone. I'm David Jenkins. I'm a medical microbiology consultant in Leicester, England, and it's my great pleasure to be the co-chair of this webinar produced by the International Society for Antimicrobial Chemotherapy. We have two fantastic speakers today, and they'll be talking about artificial intelligence and its role in infection. But before I introduce the speakers, I want to introduce my co-chair, Professor Marco Falcone. Marco.
Good morning to everyone. Thank you, David. It's a great pleasure for me to chair this meeting. And the topic is very important. Artificial intelligence, is a very, very important step to go ahead with the knowledge of science and medicine is very important. And I'm very honored to introduce two fantastic speakers. Are also two friends, but they very, very distinguished speaker on this topic. I'm an infectious disease from the University Hospital of Pisa. And David, I give you the talk to introduce the first speaker.
Right. Thank you very much. Well, when I was asked to write the preamble for this talk, I thought the natural thing to do would be to ask ChatGPT to do it for me. And this is what it came up with. Artificial intelligence can support antimicrobial treatment decisions by rapidly analyzing large volumes of patient data, laboratory results, and local resistance patterns to recommend the most effective and targeted antibiotics. By integrating real-time information from electronic health records, AI can help clinicians choose optimal treatments while avoiding unnecessary or broad-spectrum antibiotic use. Additionally, AI systems can detect emerging trends in antimicrobial resistance, allowing for early intervention and improved infection control measures. This contributes to better stewardship of existing antibiotics and helps prevent the spread of resistant pathogens in both healthcare settings and the community. So hopefully that's the two speakers will agree with the general gist there. We've got two eminent speakers who are going to provide not artificial but real and timely intelligence on how much of this is possible now, and what we may expect in the future, and how humans should use AI tools. So first of all, the first speaker is Professor Adrian Egli. He's the chair of medical microbiology and the director of Institute of Medical Microbiology at the University of Zurich, Switzerland. His research focuses on host-pathogen interaction and studying how AMR and virulence evolves. For this, he uses different omics techniques and bioinformatics and machine learning techniques for data analysis. Although Professor Egli is based in Zurich today, actually he's joining us from Istanbul, where he's collaborating with researchers over there. And I'll hand the screen over to Professor Egli now.
Thank you very, very much for the wonderful opportunity and also these kind words of introduction. It's a pleasure to give this seminar to you. Tracking antibiotic resistance with artificial intelligence is the title. And if we think about AI and AMR, I think it's very important that we consider the two problems we face by accessing, for example, data in this space. So first of all, we have different lead pathogens. So Staphylococcus aureus, Klebsiella, Escherichia coli, they all have specific characteristics, and these characteristics we need to collect and integrate into our models, and that means we need a lot of data to do that. Then there are certainly also specific sample types, so it really depends where an infection happens, if this is a bloodstream infection or if this is a urinary tract infection. This can really make a difference. I think this is the first challenge, let's say, to access the diversity of the different pathogens. But because we talk about AMR, Obviously, also what is very important to consider is different resistance mechanisms which come into play. And here, biology and nature has generated a potpourri of different options. You can see here porin laws as an example over here on the top left. Then you have efflux pump activities, target site modification, for example, in ribosomes, but also obviously in the cell wall, and then antibiotic cleaving enzymes. And all these diverse resistance mechanisms, they sometimes are based on a single gene, but very often are combinations of genes. It's not just about presence and absence of a point mutation, but for example, also really like modulation, for example, of gene expression. So a quite challenging topic to address. And the question is a bit also where to start. And I would like to start with the way the physicians interact with the lab and the different stages where AI can be placed in the diagnostic workflow. And this obviously also includes the antibiotic susceptibility testing. And this is a cycle I call the brain-to-brain cycle. So you have a physician thinking about a specific situation, a patient, for example, who's sick with fever, let's say, and coughing. And then the physician would decide, okay, I need a certain test, and orders this test in the lab. And that's the pre-analytical period. In this period, what is very important is diagnostic stewardship. So the lab could give recommendations, what test would be useful in such a situation. Maybe the physician knows on its own, but I think it's very important to kind of have this interaction with the lab in the beginning of the process to get the right test for the right situation. Then in the lab, you have a series of analytical steps. There are plenty of examples for artificial intelligence. We will see a few during the talk. And then I think something which is heavily underestimated and very, very important is the post-analytical part. We are stressing the labs a lot to have short turnaround times, which are good. Don't get me wrong. I think it's very important to be fast and efficient. But what also needs to be efficient is the communication of the result back to the physician, because here the actual action then happens. And if you do not do this, and if your message comes to the wrong person, let's say, there's no impact for the patient. So the brain-to-brain cycle is the physician thinks about an assay, sends this to the lab and gets something back to his brain and then takes action. Now, at all these steps, pre-analytics, analytics, and post-analytics, we can place artificial intelligence. And for this, I would like to start with a questionnaire. And please take your smartphone. I hope this works around the globe. You can scan the QR code. It's anonymous. And you can try to answer this question. Who has used ChatGPT or a similar situation to ask for diagnostic advice, like a pre-analytical advice? Who has used ChatGPT? Maybe I wait for 10 more seconds. Okay, very interesting. 71% said yes, 31% said no. So a lot of people use ChatGPT to receive medical advice. A word of warning here to tell you, and you will see this now in a second. ChatGPT is not a diagnostic software, and it's also not IVDR approved. In Europe, for example, you would need IVDR approval to use it. So maybe we can say here it's really an advice, so you want a second opinion and so on. But I think it's very important to validate software tools like, chatbots. Okay, we asked ourselves exactly this question: how good is ChatGPT and other large language models in providing pre-analytical advice? And what we did is a study where we took OpenAI and Meta, so that's the producer of Facebook. They have also chatbots. They are called Llama. There are different Llama versions around. Currently, the newest version is Llama 4. And in ChatGPT, it would be the ChatGPT 4.5 model. Now, what we did is we formulated 30 pre-analytical questions. So these included things like, how much blood do I need to take into a blood culture flask, or how do I exactly transport a biopsy to the lab, things like that, typical pre-analytical questions. We used then the 3 Llama 2 models, which were running in my institute on a server. And that's the advantage of Llama 2. You can run it in-house, so you can share potentially confidential information, whereas with ChatGPT and other commercial solutions, you cannot. I mean, everything you type in into the browser ends up at the company. So you have to be super careful what you share. And we wanted to compare the performance of these two. So we had 6 chatbots we compared against each other with 30 questions, so that generates 180 outputs. Now, how can you compare those? You can first of all just look at how much words do they produce. And we probably know in our own experience, ChatGPT likes to talk. And so the amount of words per answer were substantially more in the OpenAI provided tools than in the Llama 2 tools, which are marked here in blue versus red. So each colored line is a chatbot, and you see here the individual questions on the x-axis and the amount of words it provided on the y-axis. Then, I mean, that's one thing to just, observe. But then we also generated a textbook, an ideal answer. So we took the question and asked 5 experienced medical microbiologists to come up with a textbook answer. And this textbook answer was then compared to the output of each chatbot. And there are different ways how you can compare them. There are tools where you can compare the content. So, for example, are similar words appearing in both outputs? Is how similar, how close to the truth, to the ground truth, the textbook essay, the answer would be. And these essays are called, or methods are called Bleu and Rouge and BERT score. Here, I will spare you with the details, but I think it's very important to also realize again, ChatGPT was much closer to the textbook answers than the Llama 2 models. But what now is probably most important is, what are the users saying? We send out these 180 answers to a series of colleagues, and approximately 50 colleagues answered. And you can see here they judged 2 features. So for every text, they read through the answer of the chatbot and then said, is this accurate? And they could judge that with a score. And is it complete? Is the answer complete? So, I mean, accuracy means, how precise is it? And the completeness is there something important missing? And again, you can see ChatGPT much, much stronger in the performance than the Llama 2 model. I mean, nowadays, if I would redo this, obviously you would need to take the most state-of-the-art model. So this comparison is not super fair, because Llama 2 is now an outdated model. But still, I mean, it's good for us as a template. How do we compare in the future outputs of large language models? Now, what was worrisome to me is this graph. Here you can see the individual expert groups. So if we take these 50 experts, we could split them into medical microbiology with experience in training, infectious disease experts which are experienced, lab technicians, medical students, and infectious disease experts in training. And what you can see is that the score, the more away you move from the domain expert, the more likely it is that people will say, oh, yes, this is a good answer. So in terms of accuracy and completeness. And I think that's very, very important. We have to keep this in mind. If you ask a chatbot something which is not your core expertise, it's more likely that you're going to believe this. And I think these chatbots, if we use them, need a very careful evaluation. Okay, so that was the pre-analytical part. Now, analytics. There are 2 examples which I would like to share with you. The first one is about plate reading. in the workflow, you can take an image of a bacterium, and you can ask, for example, what do you see here? And I will cut this short. I mean, it's very clear this is a Staph aureus because you have learned it. I mean, it's yellowish colonies. There's a hemolysis zone around it. It's very, very likely that this is a Staphylococcus aureus. Now, AI can also do this. And what AI does is it uses convolutional neural networks to do that. So it takes an image, it extracts features from the image. So for example, the size of the colony, the color of the colony, what happens around the colony, is it shiny or not? And all these informations go into the neural network. And then based on a probabilistic categorization, you get an output with a likelihood. And in this case, you would say it's most likely a Staphylococcus aureus. Now, this works very nice on agar plates to screen if there is a colony present or not. So positive, negative plate screening works with a very high sensitivity and specificity. And this is in commercial systems also available. Identification of the bacterial species is much, much harder to do and not yet available. So if you go, for example, you take a blood agar plate and you would say, okay, what is this? I mean, the use case I showed you before, a Staph aureus, for example, it can identify that. And there's a very nice paper published in Nature Communication 2023 on urinary tract infections, but it's not yet in the commercial system available. What also works, and this is available, is if you use a screening plate, like a chromogenic agar plate, for example, and if you see a pink colony, you can almost for sure say that this is an Escherichia coli. Okay, so this is just the identification of the bacterium, and obviously a rapid identification of a bacterium might also, based with your endemic knowledge of the situation, of the local situation, you could then, for example, say, okay, very likely there's a resistance against this and this based on my experience, and then you could adapt the antibiotic therapy. But what also works is that you can measure the inhibition zones. So for example, in disk diffusion assays, and this is a paper from Geneva I show you here, you have two examples, Pseudomonas aeruginosa on the left side and Escherichia coli on the right side. Again, each column here is a measurement. Different antibiotics were measured, indicated here by the colorful line. And you can see that it was always the same bacterium measured over time. And overall, there's a very high consistency. So you can say there are a few exceptions, five exceptions here, but in general, it's very, very consistent on what it measures. And the authors here conclude that currently all results are approved by clinical microbiologists, but in the near future, these instruments would automatically deliver the SIR category without requiring a human check. Maybe not an immediate check, perhaps a later check, but I think it's very interesting. For example, if your AMR testing is finished at 2 o'clock in the morning, it could automatically, in theory, release some results to the intensive care unit, let's say, where it might make an impact potentially. But again, this needs to be studied. If delivering a result at 2 o'clock in the morning really has an impact on the patient management, because I'm also a bit doubtful there, but I think this is something, is really workflow management. Okay, so my question is, now the next question, please, again, take your smartphones and maybe we can really keep this an interactive session. Have you used ChatGPT in the past to assess an antibiotic drug resistant mechanism? Okay, most people have not. I think a few tried it out. So what I will share now with you is the ChatGPT expert, the UCAST expert. What we did in Basel, in Zurich, we asked this question. Michel Bresson, a microbiologist in training, actually was wondering, can we use disk diffusion essays and images and ask what is the underlying resistant mechanism? And what we did is in the commercial version, the paid version of ChatGPT, you can generate so-called GPT experts. How this works is you log in into your account, then you go on the GPT expert, you say, what is the task of this GPT? It's like an AI agent. And then you can upload and share specific documents. Then it knows very, very well these documents. And that's what we did with the UCAST breakpoint table, the expert rules, etc. were uploaded and thereby generated a chatbot which had the language capacity of the GPT-4 model, but was specifically trained with knowledge around UCAST. And then we uploaded, on one hand, the image, which you can see here with the disk diffusion measured, then an image, the exact same image, without these colorful circles, like the original image. And then what we also did is we uploaded the measured inhibition zone. So the number here indicates the millimeters. So this is 30 millimeters inhibition zone for meropenem. And so we had meropenem and 30 so that it knew, okay, this is what was measured. And then we uploaded this with the exact same prompt all the time. And we asked, what is the underlying resistant mechanism? Is this none? Is this ESBL? Is this AMP-C? Or is this a carbapenemase producer? And the output you can see in a second, because first we asked, again, independent human experts. They saw the exact same thing. They got the table and the image, and then we asked, what do you see here? And the sensitivity and the specificity is indicated here. We had 3 different experts. They sometimes disagreed slightly with each other, but overall, very high sensitivity, very high specificity for ESBL, AMP-C, and carbapenemases. So this is good news for the microbiologists. This is not the robot. Now comes the AI. The AI also has very high sensitivity, 95%, 96% for AMP-C, and 100% for carbapenemases. But what you see here, the red numbers, indicate that the specificity is not very good, and it overcalled resistance. And in the case of ESBL, it was very clear what happened. Klebsiella pneumoniae, as you know, has an amoxicillin intrinsic resistance, and it misinterpreted this as ESBL, despite the fact we tried to train it around and asked and told it, no, this is not an ESBL. It was a bit stubborn, I would say. The AI was stubborn and didn't consider that amoxicillin is not an ESBL. So quite interesting results, I have to say, that a multi-model commercial software, which everybody on the world has access to, can do partially the job of a well-trained medical microbiologist. So I think that's quite interesting. Clearly, the technology is not yet there that we can use this in routine diagnostics. It is not an IVDR-approved method. You cannot use this in routine diagnostics as it is. You also should not. But I think it's important that we play around with this. And I think this is a very first step going into such a direction. Now, the last example I would like to give to you is with MALDI-TOF. And MALDI-TOF, as you know, is mainly used for species identification. But a couple of years back, we asked ourselves, can MALDI-TOF also directly predict the AMR profile? As you know, there are a couple of papers out there which indicate a certain peak, which, for example, identifies in Escherichia coli STI 131, which is very commonly ESBL carriers. And we were wondering, does this also work on a larger scale? And so what we did is we generated a ginormous database, which is open source now, by the way, so you can download it here if you follow the reference. We had 300,000 MALDI-TOF spectra matched with antibiotic resistance information from VITEC2. So this was a categorical interpretation with resistant and intermediate and susceptible on the other hand. So this was before the UCAST modification that intermediate should move to the other category. I apologize, but that was as it was at that time. We then modified, so we had 4 different centers sending us data, and although they all used the same MALDI-TOF, there are differences between MALDI-TOF systems, and that's a very important thing to consider if you do these kind of work. You want to know how well does the algorithm work in the other labs. And so for this, we had to harmonize first the MALDI-TOF spectra, and we then trained the system and used 3 different machine learning models, and then predicted and compared the output. So this is the key result of this study. You see here the prediction of susceptibility with the area under the receiver operating curve. 0.5 would be flipping a coin, and 0.8 is not bad, but it's clearly not perfect. Again, this is not a result which you would use in a clinical routine, but it goes into the right direction. So you see here oxacillin resistance or susceptibility, that would indicate MRSA. Ceftriaxone resistance in Switzerland, I mean, where we have very little carbapenemases, this would indicate ESBL for both E. coli and Klebsiella. And this paper is 3 years old. That's a century in the world of machine learning. And since then, many additional new papers came out improving this algorithm. And what you can see here is the learning curve. That's also a very important output. You see here number of samples versus the performance of the model. And you can see you need to efficiently train such a system between 2,500 and maybe 7,500 isolates. So you need to pool data else. And I think it's important in certain machine learning models to also work together and share information with each other. Okay, the very last example is on sequencing. I keep this short. There's the holy grail at the moment in machine learning is to predict the minimal inhibitory concentrations from genotypes, from sequence data. And there are plenty of examples where people try this out. I'm going to share with you one which is very nicely done. As you may know, in the field of tuberculosis, obviously, genotypic interpretation of resistance is very, very en vogue and really more and more used. But there are also other examples which are listed here with the references. One thing which is a very, very nice paper is here published also about 3 years ago. They generated a public data repository. They had 67,000 genomes in there with matching resistance information. And for a series of species, there were more than 5,000 isolates. So for these bacteria which are listed here, I think the data is robust enough. The problem is the global data distribution. And this is something we have to address as a community. In this model here, there's no data from Africa, almost nothing from South America, nothing from India, and also nothing from Australia. And I think it's very important if we focus on, I mean, AMR happens in Africa, in India to a large degree. And if we do not have data access there, I think all the models we develop in high-income countries, for example, these models will likely then not necessarily work and cannot capture the biological diversity we obviously observe in Africa and India, let's say. So it's very important to work globally together as a community. Now, what they have achieved with a specific machine learning model, they achieved to predict the MIC within plus minus 1-2-fold dilution steps. And it depends a bit on which drug and which bug combination you look. The more darker green the color is, the more robust the example is. And there's still work to do, obviously, especially if you go to the certain extremes, for example, it might have some difficulties. But I think overall this is really, really promising data. Another example is Candida glabrata, just to bring here an antifungal example. What they did in this paper, they noted that a single point mutation in a specific protein of Candida glabrata resulted in a frameshift mutation. And this frameshift mutation then resulted in a destruction or in a partial destruction of this particular protein. And they used AlphaFold to calculate and predict the impact of this frameshift mutation. So you see here on the left side, the full protein, and the yellow marked part here is that part which was no longer produced in this strain. And you can see here the AlphaFold prediction without. And this is exactly where the drug binds, and this particular strain had then resistance. And I think we will see in the future more functional information also coming into these genotype-phenotype predictions where you also have structural information. Now I'm at the end of my talk. 5 key messages for you. Integration of AI into AMR is a complex topic. I think there are different areas where we can start: pre-analytics, analytics, post-analytics. At each step, high quality and careful evaluation is required. Think about potential biases. You saw the world map with these data gaps. Diagnostic lab is good to start because I think we have a high regulatory environment, maybe also a bit of a burden in terms of regulation, but it generates good quality data in general in the labs. And I think this is why laboratory medicine and radiology probably are leading the fields. There's a huge promise from image analysis to sequencing data, and we have to stay critical regarding AI. The human brain is still and will hopefully be for a long very important. If you're interested, we are organizing next week an AI course over ESCMID. Just a quick spot here, you can check it out on the website. And with this, I would like to thank you very much for the opportunity, and I'm happy to answer questions.
Amazing. That's really fascinating stuff, Professor Egli. I mean, I'm sure there's going to be loads of questions there. I've certainly got lots for you, but I'll wait until Professor Wicha has given his presentation. And that I've got really staggering opportunities and potential there. Fascinating. Over to the next speaker now. Professor Sebastian Wicha is leading the research group Clinical Pharmacy at the Institute of Pharmacy at the University of Hamburg, Germany. His research group aims to improve and individualize drug therapy by means of pharmacokinetic and pharmacodynamic and pharmacometric techniques, with a special focus on anti-infectives. He's published over 90 peer-reviewed papers in this field. His research group has expertise in bioanalysis, pharmacometric data analysis, and develops open access web-based software to foster model-informed precision dosing. Professor Wicha is an associate editor at the International Journal of Antimicrobial Agents, which is one of the ISAC journals, and served as president of the International Society of Anti-Infective Pharmacology, and also as education officer of the PK/PD study group of ESCMID. External expert for pharmacometrics for the German Federal Institute for Drugs and Medical Devices and the European Medicines Agency. So very much an expert in this field. And I'm really looking forward to your presentation, Professor Wicha. Over to you.
So yes, that was a very nice presentation by Adrian, and I'll slightly shift gears towards the topic of using models and also AI for optimizing antibiotic use with regard to dosing. And in the first part of my presentation, I will introduce model-informed precision dosing, and then in the second part, I will show you how machine learning and AI approaches can enhance this process. So I have no disclosures for this talk. And just to bring everyone up to speed, maybe you are already familiar with it. This is how the classical therapeutic drug monitoring looks like. So you give a dose of an antibiotic, and then intrinsically there is a pharmacokinetic profile, but usually we measure a trough concentration, and then this trough concentration is interpreted in relation to a therapeutic window where you have a minimal effective concentration and a minimal toxic concentration. So you want your trough concentration to be in this window. So you keep the dose if you are there, and you increase or decrease if you are outside by using usually a rule of thumb or some nomogram. So with model-informed precision dosing, you can directly adjust to this workflow, but instead of ignoring the pharmacokinetic profile, you actually use a pharmacokinetic model or a population pharmacokinetic model to interpret the observed concentration. And that does not have to be a trough concentration. It can be actually any timed concentration which can feed into these models. And then we will see how with these sparse data, you can actually parameterize a full PK curve. And then instead of just saying, oh, okay, we're good, you can then ask the model to predict a dose for a specific target concentration. And then this model will tell you, okay, you have to give this or that dose or this or that dosing interval to reach this very target then. So you can aim and optimize. And of course, this target Does not have to be a trough concentration, but it can be also something more complex, such as the AUC, for example, or time above MIC that you usually cannot calculate directly from a trough, for example. So to do that, there are a number of software packages available, academic developments, commercial developments, which are all very easy to use. This is an overview that our colleagues in Belgium did a few years ago. And this is a software, this is an open access software that we develop in our institution since the past 10 years. So we are always adding new functions and drug modules to trying to disseminate the results of our research. So as an example of such a software, I'm showing you very quickly the TDMX software. As I said, there are many other softwares as well, and the user interface, of course, looks slightly different usually, but the functionality is often very similar. So here in TDMX, here on the left-hand side, you have a panel where patient data can be entered. For instance, here, patient covariates. If the patient already got a dosing regimen, this can be added here under this dosing tab. And then the predictions, how this predicted dosing scheme would look like in this patient is outlined here. And then there's here some tabular information. And here, for instance, this is the example of vancomycin. You also get the AUC over MIC ratios that are calculated here in each dosing interval. And then you see here three different things. You see the population prediction in blue, and you will notice these have wider prediction intervals. So here, this is the prediction for a patient with these specific covariates. But here in the population prediction, the drug measurement is not yet considered. And then in orange, you have the drug measurement, and then you see that it's deviating a little bit, that curve, from this population prediction, and also the prediction intervals become more narrow. And that's what those softwares do in the background. They balance the individual information coming from this sample with the population information that is stored in the population pharmacokinetic model using a Bayesian estimation approach. And through that approach, it's actually possible to estimate an entire PK curve from just very sparse data. And of course, if you want to make that work, this process, these models need to be externally validated. And this is something that we have done in our group quite a bit in the past years, that we try to systematically recreate models from the literature and then run them on external data sets. So these are just 3 papers here for vancomycin, where those models that were identified here were then implemented in the TDMX software. And we even took it one step further, and instead of using a single model, we also have developed so-called multi-model or model averaging approaches. How do they work? So here you have a patient in your ward, and instead of fitting a single model, you can fit a number of candidate models, which is illustrated in this cartoon here. And then you may have some models that describe this individual better than others, and those models then get a higher weight, and models with a not so well model fit get a low weight here in this weighting step. And then the averaging comes into place. So you average across those individual predictions using these weights, which is this black line here. And then you consider all these models together, but you weigh up or you weigh down their importance within this individual patient. And the other approach would be an individual selection approach, where the winner takes it all and the other models would be discarded. And this algorithm actually works pretty well. This is the example where we first published this algorithm for vancomycin. There are now a number of other indications available as well, where this algorithm also worked. So you have here bias, RMSE, and then here different parts of the data set that were provided. And you can see the individual models here. They tend to show some positive or negative bias and some imprecision, but here in black, if you use those models together in the model-informed precision dosing workflow, you can get unbiased results, and also the imprecision is usually in the lower range of the best performing individual models. And this is available also in our software. So if you would hover over model averaging, you can use this algorithm also for dose calculations. So in that first introduction, I wanted to show you a little bit what is the state of the art, what is possible with conventional modeling techniques. And now in the second part, I will try to show you how AI and machine learning approaches can potentially help to further improve this model-informed precision dosing workflow. Therefore, let's quickly compare the properties of the Bayesian approach and machine learning approaches. So actually, to develop such a population PK model, you do not need too much data. So the required sample size is not too high. So with some 50 or 100 patients, you can actually probably develop already a very decent model. The advantage of the conventional Bayesian approaches is that they are interpretable by simulation. As you have seen in those plots, you can generate those PK curves. So if something funny is happening, you usually will see that. Predictive performance is usually high, but that cannot be conclusively answered yet because we have not so many examples here on the right-hand side. Probably in the end, we will see how that race will go. Also, need for adherence to sampling schemes is low. You can use any timed sample in a Bayesian approach, and the computational intensity, well, is medium. If you now compare that to a pure machine learning model, we have seen that also in Adrian's examples, you need a lot of data to parameterize these models. So you would need much higher sample sizes here. And then the pure machine learning models are also not interpretable by simulation. So you will get back one number and you will have to believe it, that this is the dose. You will usually not see a graph, for example. And also you will need to strictly adhere to sampling schemes. So if the model was trained on trough concentration or on specific time points, it may not extrapolate well if you go a bit to the left and to the right to other time points. But they are very efficient. So since we have usually not these very dense and high data sets available, we rather see at the moment those hybrid approaches where conventional population PK Bayesian approaches are combined with machine learning examples to kind of try to get the best out of both worlds. And here I want to show you some examples. And one example here is on ganciclovir and valganciclovir, a very nice study by our colleagues in Limoges. But here you can already see the high demand for data of these machine learning algorithms. So they actually had 10,800 patients, and those are difficult to recruit. So they actually used the population PK model in the first step to simulate from several population PK models those virtual patients with a very dense sampling scheme, and they simulated true AUCs. And then on top of this synthetic data, they estimated a machine learning algorithm to predict the AUC with several limited sampling strategies. And then they found two optimal sampling strategies and then evaluated this algorithm. And indeed, here in the external data set, they could show that this learning algorithm was actually superior over using single Bayesian population PK algorithms. Or another example that's not from the field of anti-infectives, but that is in cancer therapy, where it was about looking at the time course of neutrophils after chemotherapy, so which is shown here. And there also you usually have only a few time points where those neutrophil counts here in green are being measured. So also here, they used the raw data and fitted a PK/PD model to it. So they augmented this raw data and then provided both the real-world data, the data augmentation, and also some features of the PK/PD model. So some model parameters to a machine learning algorithm. And then they compared the predictive performance to classify this neutropenia again with different approaches, either using only machine learning, PK/PD enriched, or then the full set that I just described. And also here, it was apparent from their evaluation results that, well, the PK/PD alone did not perform that well. Also, the machine learning alone did not perform so well, but in the end, the enriched or the augmented approaches, where both approaches were used hand in hand, actually did perform best. And one more example where we are again in the field of model averaging. Here, this is also vancomycin, and the authors here used exactly the same data set that we used to develop the model averaging algorithm. So we worked together here. And since I showed you in the model averaging algorithm, you need TDM data to be available so that the model learns about the individual patient. Here, the authors wanted to take this a step further and wanted to see whether it's actually possible to upfront use a machine learning algorithm to learn from, for example, the patient covariates, to even come up with a weighted prediction before the gift of the first dose. So kind of, if you know that you have a patient with obesity, for instance, that the machine learning algorithm would learn this from the data. So obesity model is upweighted. And indeed, this algorithm particularly improved the performance a priori, whereas if you have the data, then the performance-based model averaging here or the synthetic model combination performed very similarly. And one more example, also a very nice one, where machine learning helped to improve the performance, is a so-called flattening of the priors, where you kind of tune down the Bayesian prior in the estimation process. So you tell the model to listen more to the data. That would be here in this case, that you fit more to the individual data points. So you balance, you change the balance towards the data. And then the authors here around Jasmine Hughes use the machine learning algorithm to tell the algorithm when it makes sense to flatten the prior in the Bayesian estimation. And also here, they could show that this could lead to an optimized predictive performance. One example from us. So sometimes you have to deal with unstable patients, for example. So that's something that you can see here, that your PK prediction deteriorates. Well, before you embark in machine learning, you could actually first do some practical checks. So for example, did you consider time-varying covariates, such as a change in renal function? Maybe that explains your deterioration here. Or maybe if you have another model, check that if that is available. Or a very crude approach could also be to consider removing older data, which I'm showing here and flipping back and forth. And that may also help to improve the predictive performance of the future if for some reason the patient has changed substantially, and that is not considered in one of the models. But what is very important in MIPD is the paradigm of garbage in equals garbage out. So if you have low quality data, you will not have precision doses, but you will have also very imprecise doses. And that means you have to take care of the dosing time, administered dose, infusion length, timing of the TDM sample, and also the covariates, that they are adequately documented. And this is particularly important if you want to use this model to also, this data to also build a model on top of it. So this is quite challenging at the moment, that all these timings are being accurately documented. But I think the future is bright. So in addition to the conventional chromatographic and immunoassays that we're using at the moment, when we go through this dose adjustment cycle, some biosensors are actually around the corner. You may know those biosensors from diabetes care. Those are also developed to measure drug concentrations, antibiotic concentrations in the interstitial fluid. And as you can imagine, those sensors can measure very, very frequently. Every few seconds, you will have a data point. This is some example here for vancomycin, where this sensor was used to measure vancomycin exposure in rats. And you can see those very, very dense data that can be obtained here. And I'm very convinced that we'll see the applications of those biosensors in humans very soon. And I'm also convinced that machine learning will be extremely interesting to be used on this data and will help us a lot to overcome some of the gaps we have at the moment with regard to documentation and the very sparse data situation we have for therapeutic drug monitoring. So to wrap up, many things are already possible as of today. So model-informed precision dosing is very powerful and can streamline dosing decisions. So you can ask those models to calculate a first dose. You don't need to wait for steady state. You can use any timed sample to interpret the PK curve, and with a single sample, you can estimate the full curve. And there are some nice examples where those machine learning approaches can help to enhance the MIPD performance. And then those biosensors may really unlock the full potential of machine learning-based approaches. And this will be very interesting to see more in the future. And of course, what is also important is that we need also clinical trials comparing individualized approaches versus standard dosing. If you want to read more about model-informed precision dosing, a short advertisement here. Together with some colleagues, we published this paper where we both went into the state of the art on the technical end, but we also looked into clinical applications of MIPD and where the evidence is. And with that, I would like to thank you for your attention. Thanks for listening, and also thank my wonderful team here in Hamburg and our sponsors that make our work possible. And I'm looking forward to the discussion.
Thank you, Sebastian. Thank you, Adrian. Two really, really spectacular lectures. Then we have a lot of questions from the audience, and I'm reading the chat. We have a few minutes. Then I ask just you very brief answers to this question. First question is by our colleague. I'm told that the AI models use a huge amount of energy and have a large carbon footprint. I don't know if you, Adrian, do you have any thoughts or comments on the environmental impact of AI?
Yes, that's a very good and important question. There are a series of Nature articles and news articles which very nicely summarize this issue. I would highly refer to look there. Just a quick thing. At the moment in the US, 7 terawatt hours are used for AI. This will increase over the next 5 years by 100-fold. 1 terawatt hours is what a nuclear power plant produces. So we will run into energy dependence massively with the increasing use of AI. Now, the good news is that more and more efficient models come out, which calculate more efficiently. But still, the demand will also increase. So I think this is a very, very important topic to address. Very, very, very important.
Thank you, Adrian. Another question.
Maybe can I just add one thing? To charge your laptop, because everybody has a laptop, you need 100 kilowatt hours. To generate an image with AI, you need about half of it. So every time you think you make a funny picture with ChatGPT, it is half charging your laptop, just to give a bit of a relationship.
Very good, very good, very good explanation. There is another question for you. What is the advantage of UCAS GPT Expert over, for example, antibiotic created by MSF a while ago?
Not really an advantage. I just looked up the tool when I saw the question in, it's amazing what they did from Médecins Sans Frontières. Really, really nice. It looks fantastic. I will certainly try this out. Our purpose was to use a multimodal large language tool, which everybody has available, and we were more playing around of that. I have not compared the performance of these two tools or against each other, but it's great that this exists.
Can I
David,
ask
do you have any question? Yeah.
Yeah, can I ask a question to Professor Wicha? Your model, pharmacokinetic modeling and predicting depends very much on collecting specimens from the patient, at least on the first dose. But our turnaround time for for many drugs for therapeutic drug monitoring, apart from perhaps gentamicin, vancomycin, it tends to be quite long, maybe in the order of days, and isn't well suited to your modeling. What is, you mentioned biosensors. Do you see that as a major stumbling block before these models can be implemented?
Yeah, you're fully right. This can be an issue if the hospital has no access to these assays, or if the turnaround times are very slow, several days. It is not very much useful in the case of anti-infectives, because there you usually need those answers quickly. So if you want to implement MIPD, it is it should go hand in hand with implementation of bioanalytical assays within the hospital. Right. So you have short turnaround times. The biosensors may be of much help there because they provide the signal in real time. And I think also there's development, for instance, in the direction of lateral flow assays, for example, similar to those COVID tests. They can also be semi-quantitatively interpreted. I've seen some examples where the quantification actually went really well. So this is also research. This is not implemented yet, but this could be also an alternative if you don't want to buy a mass spec or an HPLC, but a small handheld device where you can also measure the drug concentrations then. Yeah, but it's a very important point.
Yeah, we've got another question from Martin Goldberg saying, using modeling to calculate how much of a drug to administer to achieve a target assumes that the mechanisms for excreting and degrading the drug are saturated. If not, adding more drug may not give the expected concentrations at time X post-administration. Different people have different rates of excretion, degradation, etc. Makes modeling much more complicated. I guess that's true. Needs a huge number of data sets to make the modules robust and accurate. But I'm sure you acknowledge that.
Yeah, that's a very good point. So maybe the first comment on the saturation, that's very much true for classical therapeutic drug monitoring. For model-informed precision dosing, this doesn't have to be the case. So there you can also work with non-steady state situations because the model can account for this. And then, yeah, of course, different people have different rates of excretion, degradation. Yes, that's very much true. Sometimes we do have covariates for this, renal function measurements, for instance, that we can also implement or that are implemented in the model. But yeah, there is a point, of course, that sometimes things happen and we don't have a covariate for this, and the PK goes in one or the other direction, and it's not a covariate in the model. And for that, I think we can particularly leverage machine learning algorithms that can detect those patterns and maybe help us to improve the predictions.
And can I ask a question aimed at both of you, actually, which is, do you see the work you're doing aimed mostly at patient outcomes or preventing antimicrobial resistance, or is it a combination of both?
Yeah, for me, it would be a combination of both, I would say, because those PK/PD targets that we aim for are, of course, usually related to efficacy, also minimizing safety. So that's clearly the patient outcomes perspective. And for some drugs, we also have an understanding of targets that prevent resistance development, for example.
Professor Egli?
Yeah, also both. But I think what we need is, when we have a diagnostic device, we need careful evaluation of the clinical impact. And what is currently missing are randomized controlled trials, clearly. All the data I have shown you, and 98%, I would say, of the publications are retrospective analysis of those tools. So we need prospective randomized controlled trials. We need trials of AI tools across with really independent validation. I saw this year at ESCMID, there were fantastic talks, but everything was retrospective and never validated with another center. So please take your tool and place it somewhere else and see how well does it performs. That's very, very important.
And just one last question. I mean, there's loads of questions, but we're running out of time. But just one last question to both of you. Is this the death knell of infection experts, or will we still have jobs to do?
So I also saw some worried and said like, oh, I'm not sure about AI with my job. We have to adapt. It's a new technology.
Yeah.
I mean, every time when Multitoph came out, everybody was worried that nobody learns anymore the biochemical reaction tables, you know, to identify bacteria. Yes, we need to take this serious, but it will not happen tomorrow or in a year. So I'm optimistic that I will still keep my job.
Excellent.
Yeah, I would agree to
Sebastian?
that. I would agree to that. I think expert knowledge is still very valuable to interpret the results from these models, particularly for the MIPD part. And then it's a new technology and it will make things more efficient, which we all hope, but it probably will take some time to replace us, and that's very unclear at the moment.
So we'll still have jobs, but they may not be the same jobs, possibly. Yeah. Brilliant. Thanks very much indeed. I'll just hand over to Marco now to wind up for today's session. Marco.
Okay. Thank you. Thank you very much to everyone for this fantastic session. It confirms that ISAC webinars are really, really interesting. We had a lot of participants, and you will find out also this session on the ISAC website. You will find this meeting, and you can also share this meeting with your colleagues. Then we are out of time, and it's the time to close. Thank you very much, Adrian, Sebastian, David, and we see to the next ISAC webinar. Bye-bye.
Bye-bye. Thank you very much, both.
Bye-bye. Thank you.
Thank you.
Thanks.